What does one house say to the other? House it's going. What do the dolphins shout when you got stuck in seaweed? Help! Yeah, very good. And where did the whale play his violin? In the orchestra. We're going to say, well, whales aren't orcas. Like a 7-year-old that wrote the joke, so don't overly criticize. Okay. How we doing? How's everybody feeling? Good? Okay. We ready? We ready to shift gears into sort of some heavy wound healing stuff? Did you guys take a look at the updated lecture? We only have 84 slides to go through today. I'm just going to hold the button down. No, I'm just kidding. Now, today and Monday. So I updated the slide deck because there's some new regenerative medicine stuff that I wanted to add in that I've been holding off for a little while, but I think it's about time to present it to you because there's a lot going on in the field. So today we're starting tissue repair. You've got a post quiz on Friday, or it's open now, but it's due by Friday. And you should expect some of this wound healing stuff to show up on the post quiz. And then we'll finish out wound healing, or the fibrosis and healing part. We'll finish what we don't cover today. We'll finish out on Monday. Okay, so we're right on schedule. Hopefully everything's going smooth. It seems like DB Learn style. People are getting used to it. The number of announcements is going to start to, settle down in the beginning of the semester, is reminding you, what time class starts, and make sure you have a pre and post quiz. We've got our SI session times and dates here, so hopefully all of you are taking advantage of all these opportunities. Okay? Any questions before we get started? All right. Excellent. Well, if you took a look or a preview at the slide deck, you'll see that there are some gory pictures towards the end. You guys take a look at those? Okay. So we'll get some warnings ahead of time, and hopefully it won't be too bad. But in order to get started, let's look at this graph. Now, we've seen this graph in previous lectures. We've talked about aspects of this graph. It's a new graph, but it's characterizing the same material as what we've seen in the past. So if you're looking at this and you're saying, oh, it doesn't look the same, that's right, it's not the identical graph. If you're saying, wow, that looks very familiar, well, it's very similar to kind of the James Anderson acute inflammation slides that we compared and contrasted to. So this is actually from a textbook in the 90s. I think the James Anderson was like 1988. This is 1996. So this is much more modern day, right? Some of you are laughing because it's still the last millennial. But this is from Clark et al. And what they're characterizing is this wound healing response, days on the x-axis, and on the y-axis is some relative amount of the response. And letter A is identifying inflammation. So you could write inflammation above A, letter A. So you see how A is right here, and there's an early and a late phase. So there's an early and a late phase of inflammation. What do you think the early phase is characterized? What cell types do you think migrate in the early phase of inflammation? Neutrophils, very good. That's another way to ask the same question that I've said now 17 times, I think, which is what are the first responders, right? Neutrophils. So you can write that these are neutrophil infiltration, and then the late phase of inflammation, not the late phase of inflammation. The late phase of inflammation, what cell types do you think are responsible for that phase? Macrophages, very good. Okay, they're the second on the scene. And we've been abbreviating macrophage with this symbol. For those of you that are coming for the first time, welcome, right? So this is our macrophage symbol. And then you can see in portion B, B is granulation tissue formation. Granulation tissue formation is another terminology for these macrophages make a lot of these chemical mediators. And we looked at that. And there's a couple of chemical mediators that stimulate new blood vessel development, particularly one known as vascular endothelial growth factor, VEGF. And so VEGF causes new blood vessels to form, but they're very small little vessels. They're the capillary beds. And because in this next phase, this next peak, we have a rich vascular tissue that has all these small little vessels. It has a granule-like appearance, and that's called granulation tissue. So the macrophages come up and they come down, they release all their chemical mediators, they stimulate new blood vessel formation, and now you get a granular-looking tissue because it's highly vascularized, because the macrophages stimulated new blood vessels to grow in. Make sense? So granulation tissue, highly vascularized. And then you can see in portion C, we get the wounds that have been starting to contract, kind of finish their contraction in C, and we start releasing collagen deposition from fibroblasts that migrate in. Fibroblasts are skin cells. Let's pretend we're talking about a skin wound. These skin cells from a wound migrate in, and they release copious amounts of collagen. Now, if this wound wasn't on the skin, it was in the heart or in the liver or in kidney, you would still have tissue fibroblasts. They just wouldn't be skin fibroblasts. They would be fibroblasts of that particular tissue bed. And they're still going to make collagen. They might make a different isoform of collagen, right? There's about 20-some different isoforms or variations of collagen. And so they might make a slightly different collagen, but they're still, unless you're going to follow the same paradigm. And they lay down this new matrix. And that new matrix that is laid down and is organized is not in the same type of organization as the original tissue. And therefore, it creates an area that is unique, and it's more fibrotic-like because it's a different type of structure, and that's what we call a scar. And you can see that on your skin, and you can see that scar in a heart. We would call it a coagulative necrotic scar, right? Or you can see it in a liver. Again, it would be a coagulative type of scar. So the other analogy would be, how many of you, put the door all the way open and it goes through the drywall, right? I mean, come on, all of us. I blame it on the kids, but I actually did it. I'm just kidding. So you can patch that, right? You get spackle and you can cover it, and then you can repaint it. But guess what? You can still see where it was, right? Because that original drywall, even though you've patched over it, you can tell where that new material went down. That's exactly like what happens with this process of wound healing. So we think of wound healing classically in the skin, but this applies to a lot of these other tissues and organs that we're going to be looking at throughout the semester. So we'll use the skin as our primary model because it's the easiest. It's one that we see, right? We come in contact with it, we can appreciate it. It also happens to be the one that I do most of my research on, okay? So that makes it kind of convenient. So collagen accumulation in this tissue remodeling phase, and you can see the resolution of inflammation starts at phase D. And phase D kind of starts here at granulation tissue, and it continues. And then last time we were together on Monday, we talked about how long this process actually remodels for. How long is a wound actively remodeling before it's pretty much quiescent? You guys remember that? Yeah, it's about 365 days or 360 days, about a year. Okay, so you can influence that remodeling in that year period of time. And many of you can remember that. You have a wound and it closes, it stays kind of red and pink for a long time, right? And so you're seeing for a few months that it's still kind of red and pink. And then finally that kind of subsides, but it's still, bless you, it's still remodeling underneath that deep tissue. Okay, so what I want you guys to compare and contrast right now is the difference between resolution and repair. Okay? So pair up, one of you is going to explain to the other the difference between wound resolution and wound repair. And then the other one's going to explain the difference between healing by first intention and healing by second intention. Okay? So this is the point at which you actually have to become social. Turn to somebody near you. Okay? And then you have to, I mean, you can sign if you're signing, and that's why there's not much length. Audible, that's great, but I'm guessing most of you need to communicate with words. There you go. There we go. Okay. In a brave soul, tell us the difference between resolution and repair. It could be person A or person B. If your partner told you a great definition, go ahead and take credit for it.
I think resolution is associated with like the return of function of the tissue, whereas repair is like loss of function, like scar that doesn't have hair growing from it, for example.
Yeah, no, that's great definition. So just to repeat, for the video and also for those in the back. So resolution is the return of normal function. So wound resolution means that all the function comes back of that particular tissue. Now, let's not use the skin as an example because that's sort of the easy one. Let's pretend for a moment that you have liver trauma or liver damage from either a car accident or it could be from chemicals. But wound resolution happens, you get a complete remodeling of that liver, and it returns back to normal hepatocyte function, where they're able to produce proteins, right? The liver is able to detoxify, and it does all of the normal activities that it is responsible for. That would be a restoration of function, and that actually happens. The liver is a very regenerative tissue, okay? And we're going to learn about that later. If you do liver damage, there's a point at which you can regenerate and have wound resolution, okay? So that's perfect. And then in the case of skin, another example would be some of the functions of skin, where if it still serves that barrier function if there's a scar, but maybe it's lacking apocrine sweat glands, or maybe it's lacking hair, follicle shafts. So it doesn't have the same makeup as it originally did, and so now you don't have wound resolution, not returning to normal function. And then healing or repair are synonymous, and so in the healing or the repair scenario, you fill the damaged tissue with new matrix, typically collagen. But again, it's kind of like that spackle on the wall to cover up the hole from a doorknob. It's obvious. It doesn't look like it used to look. It doesn't function like it used to function. Okay? Really good. Okay, how about healing by first intention versus healing by second intention? Right here.
First intention is a clean cut wound, a surgical wound, or not wound, I guess, an intentional.
It's a wound, yeah, it's still a
Yeah.
wound, yeah.
And then a second intention may be an infection occurring, chronic inflammation, different types of cells healing.
Okay, and so first intention is absolutely correct, you nailed it. So healing by first intention would be usually a surgical incision where it's a clean scalpel blade and you've got tissues that nicely approximate. What that means is you can see where those wound margins are and you can bring it back together and you can line it up. Okay? That's wound approximation. It approximates really well. In contrast, just to modify your second intention often does have infections, but really what it's referring to is where you don't have nice wound margins. Usually like trauma, where there's not a clean incision where you can approximate the wound margin. There's a giant gap of no tissue. That would be healing by second intention. Which one has better aesthetics outcomes typically? First intention. Okay, so let's talk a little bit about plastic surgery. So if you had trauma, right, and let's pretend that you had like a divot, second intention, and this filled in, and it filled in with scar tissue that's pretty obviously different than the surrounding tissue. How could one take this second intention and convert it to first, is the question. Any ideas? Cut it all out. We wouldn't necessarily staple it, but cut it all out. I understand what you mean. Approximate the tissue, and then probably very methodically use small diameter suture, small diameter bore size needle, lots of small sutures. Probably, depending upon the size of the wound, you're talking hundreds of actual suturing, right? So then you come in here, that's exactly right, and you make these incisions, and you bring it together, and now you have more of a first intention healing. Does that make sense? So that's kind of how, in a nutshell, it's a very, very brief overview. Don't pretend you're plastic surgeons and go out there and start changing the world yet. You still have medical school you got to go to, but this is essentially how this works, okay? So let's look at this chart. We're talking about normal homeostatic situation at the very top of a cell, and then we see, Shazam, there's this injury, right, that lightning bolt. And we can go one of two directions. We can go to the left, your left, and we can see that there are tissues that have the capability of complete regeneration. And so things like the epidermis, the GI tract, the hematopoietic system, as well as even in some cases we've got, well, they call it compensatory growth, liver and kidney, but it's skin, GI tract, epithelium, and the blood system. We can completely regenerate those tissues because they have renewing capabilities. Stable tissues, you lose some of it, and it can regenerate compensatory-wise. Lots of regenerative capabilities in liver and kidney, like what we talked about. And then over on the right side, you can see this is regeneration. Another name for that would be resolution. And over here, we have wound healing, or what we call wound repair. So you'll see these terminologies used interchangeably. Regeneration and resolution, and repair and healing. So repair and healing, there is a scar. We don't have normal function. You can kind of see that there's a wound, and we heal it with deposition of new collagen, just like what we've drawn right here, but you have a fibrotic scar. So scar is another word for fibrosis, right? Fibrotic scar or fibrosis is the same thing as a scar. Same terminologies that's used like in a heart attack when we say, well, I hope that, you know, grandpa doesn't have too much scarring from the heart attack. That's what they're talking about right there. In circumstances of chronic inflammation that we just learned about, we can also lead to significant tissue fibrosis as a result of this inflammatory burden that's chronic over time. All right, so let's look at a review question. What's the role of the fibroblast during the reconstructive phase of wound healing by second intention? Okay, another terminology for that reconstructive phase is going to be what? Remodeling, tissue remodeling. That was category C on that first graph. It said tissue remodeling, so that's the same thing as reconstructive phase. Okay, you're rebuilding, you're remodeling. So the fibroblast, what does it do? Does it generate new capillaries from vascular endothelial cells? Does it help to establish connections between neighboring cells and contract their fibers? Does it synthesize and secrete collagen and connective tissue proteins? Or does it provide enzymes that debride the wound of dead cells? Or two of the above. What does the word debride mean? Or debride? It means to clean. Okay? So you can debride a wound either mechanically, chemically, right? So you can scrape away dead tissue. You can flush it with a saline. Okay? So what is the role of the fibroblasts by second intention healing? Two of these? Which two? Oh, I guess. How many of you think two of these? Okay. How many think you've got the two that you want to share?
B and C.
B and C. Why? Explain. Establish connection between neighboring cells and contracted fibers. Synthesize and secrete collagen and the connective tissue proteins.
Prepare for filling the gap and
Okay.
let it remodel after.
Prepare, we fill the gap, and then we allow it to remodel afterward. Yeah, so B and C are the correct answer, and they're somewhat related to each other. C, synthesizing and secreting collagen and connective tissue proteins, that actually helps with the contraction phase or wound contraction. So the correct answer is B and C. So, I mean, some of this is, we're teaching you, but I'm showing you how I would ask this like on a quiz or on an exam question, okay? All right. This slide is characterizing hepatocytes or hepatic tissue, liver tissue. And you can see up top on the upper left, there's a normal hepatic globule. And we've got a box that is blown up to the right. So this little box right here is blown up in higher magnification, showing you the portal triad, just the architecture of the vein, the hepatic artery, and then the bile duct. So the vein is in blue, hepatic artery in red, and the bile duct in green. And it's just to give you a frame of reference, something to compare the subsequent pictures to. So in the example that I said earlier of liver damage, let's say there's an injury to the liver. There's an injury to the cells only. You go down the left pathway. So you can see that here's our portal triad, just like what we saw here, just to give you a frame of reference. And you can kind of see that this architecture, the patterning is still there, but there's a lot of cells that have disappeared because there's some sort of injury to the cells in the liver. Let's pretend for a moment that it's a chemical injury. Let's pretend that it's alcohol, okay, in an alcoholic patient. Or maybe they're not alcoholic, but they're utilizing alcohol on a very regular basis, okay? What's the difference? I don't know, it sounded better the second time, okay? So if you remove the injury and you don't have damage to the architecture, to the matrix, you can actually restore complete regeneration of that liver tissue. And that's exactly what can happen. Now over on the right side, if you do so much trauma that you stimulate a chronic inflammatory response and you bring in tissue macrophages and you start creating fibrosis in that liver tissue, when you repopulate it with cells, it's very disruptive. And so the take-home message of this particular slide is the following. If you have tissue and you just do cellular damage, you have the ability to repair. If you have tissue and you have matrix and the cells that are damaged, so the matrix and the architecture, the cells and the architecture, which is the matrix that they live in, are disrupted, you're going to have a much more likely of a situation that you're going to have incomplete resolution. So you'll have a scar, a fibrotic response. More damage when you damage the matrix plus the cells than when you damage just the cells alone. Make sense? And so later in the semester, when we look at liver and kidney diseases, we'll kind of come back to this concept where there is a point at which the liver is regenerative, and then there's a point of no return. And that delineation is where we refer to it as the liver is now cirrhotic. Like a cirrhotic liver means that it's chronically inflamed over a long period of time, and the matrix and the cells are damaged, and so you're not going to regenerate the liver. You'll have to transplant the organ. Okay? Okay. Let's talk a little mechanistically now. So hopefully you guys are getting a little bit of the patterning, right? We do a little bit of introductory stuff to kind of get you interested. Then we dive into the really boring stuff, which is the mechanism, right? And then we kind of bring it back at the end with maybe some clinical examples or some translational examples. So that's kind of, I'm just giving you the whole secret of my lecture series. Right, so the boring stuff's right in the middle, and here we go. You ready? So I want to flash back to cell cycle landmarks. And cell cycle landmarks are important for us to think about because all of this regenerative capability or this healing capability in tissues is dependent upon how the cells behave. And whether or not you can take them from a resting state, okay, like a G0 state, get them into the cell cycle to start duplication and start dividing and spitting out additional cells. And fibroblasts do this. Right? We do this in my lab. We grow fibroblasts all the time. They proliferate like crazy in the dish, and they're a great model. They're an absolutely great model. We primarily use skin fibroblasts because they're indicative of how the skin's going to heal, and we do a lot of skin wound healing studies in my lab. So, but most cells that are going to have all cells that have the ability to contribute to a healing response are going to go through some sort of cell cycle initiation. And so this is the gap 0 phase, right, where it's kind of in rest. It enters into G1, moves into a checkpoint to make sure that the DNA is legit, there's no errors. Then it goes into chromosome duplication in the S phase. Then it goes into a gap 2 phase. Now the gap 2 phase stimulates another checkpoint. That's the purpose of those gap phases, is a checkpoint to make sure that there was no problem when you duplicated the DNA. Then we go into the M phase, or mitosis, and you pull these two cells apart. I'll show you a video of this. And then you have cell division, where you've got one cell becoming two. And we have the ability, our tissues, our biology has the ability to influence every single aspect of this cell cycle. Okay? And I'm going to flash forward a little bit to some of the technologies that are being utilized today in this space of regenerative medicine. And we'll get to this on Monday, but you hear all sorts of conversations about like stem cell therapies, right? Pretty rampant. And so we'll talk a little bit about what that is on Monday, how is it working, and that's the stuff that I've added. Because over the years, this is like a new field that's been, born, and now a lot of patients are seeking out, how do I modify this to happen in my damaged tissues? Like in my knee joints, right? In chronic wounds that aren't healing on my skin. Or maybe in bone fractures. And maybe in spinal defects of the vertebral column, not necessarily the central nervous system, okay? So all of this has its roots in cell cycle division, so it's important that you understand this. The other thing I'm going to flash forward to is even further in the class is, you see these checkpoints where we pause to evaluate the integrity of the DNA? We have a cancer section coming up, and one of the things that cancer does is it eliminates maybe some of these checkpoints, where you don't pause to see if the DNA is actually sound, or if there's an error. Or we just speed this whole process up, and we, in cancer cells, have this mitotic signal just turned on like crazy. And that's why cancer grows so quickly, is because the M phase, a lot of these cells are kind of stuck in the mitotic phase. And they are replicating again and again and again, and there's no pause for a transcription check, or the integrity of, did I make a transposition error? Is there a base pair that's out of sequence? So this slide is here to give you a reminder of what you learned about in general bio is going to come back into this class and is relatively important. Now, there's a couple of different ways that cells will communicate and way that they signal. And the way that they do this is through intracellular signaling, cell-to-cell communication, okay? And so we can look at autocrine, paracrine, and endocrine. So autocrine, this is a prefix, auto meaning self. So the example is, you can have a cell that releases a substance, it binds to a receptor on itself, and causes some sort of change or physiology to take place. Paracrine signaling. Paracrine signaling looks like this cell releases a secretory agent and binds to a nearby cell, and then this stimulates this cell to do something. We looked at this in a previous lecture, and we called it, in the cell injury lecture, we called autocrine autophagy, and we called paracrine what? Heterophagy. So there's a slide called auto versus heterophagy, which is basically just an example of autocrine and paracrine signaling. Heterophagy being paracrine, and autophagy being autocrine, sorry. Then the last one is endocrine signaling. Endocrine signaling, and we'll look at this in our endocrine disorders later in the semester. We release a substance into the bloodstream, it's carried to a downstream target tissue, and it binds to a receptor at a distant location, and that's called endocrine. And so all of these things are in play when you get a cut. When you traumatize the skin, there are local tissue responses that are autocrine activity, there are paracrine activities, and then of course there's endocrine activities where you're recruiting cells out of the bloodstream. Make sense? What do the cells need to recruit out of the bloodstream primarily? Our monocytes that turn into what? Macrophages. Okay. So we're in the tissue. We have a vascular supply right here. We've got an epithelium, right, at the level of the skin. And so there's a couple of areas that are blown up here. So this part right here is blown up. This is the wall of the blood vessel, and this is its architecture. And then this point is blown up over here, and this is a fibroblast that's out in the tissue and how it interfaces with its matrix, right? So these fibroblasts, they don't just hang in space. They actually have to attach to something. So when we think about tissues and we think about cells, I like to use the analogy of, think about the old school jungle gyms, right? A lot of them, they're like this dome structure. They're made out of metal, right? None of this plastic stuff, no like ground up tires, to save your fall. I mean, it was like, I think ground glass, as my playground was, or something like that. If you fell off, you just, you're done. Just trash. Just leave them there. His brothers will grow up and take on his name, right? So you guys remember the dome structure? So, and then the kids would play in the jungle gym and they'd kind of climb around. That's how cells behave, is they have to have some framework to interact. And so this does a really nice job, this slide does a really nice job of showcasing this scaffold right here, which is the jungle gym, and this fibroblasts and how they actually attach. And they attach via integrins. They interact with their environment with these integrin molecules. So we've got all sorts of things that could be happening here, right, in this particular slide. We've already talked about what happens at the blood interface, where we're recruiting monocytes neutrophils come out first, but then we recruit monocytes out into the tissue, and they migrate to the target tissue of interest where the injury is. Well, at that injury location, we also have all of this activity that the fibroblasts and the matrix that interacts with that fibroblast can do. And so those fibroblasts will crawl, like kids crawl on that jungle gym, to get to that location, and they're going to go through the cell cycle, and they're going to replicate and make more fibroblasts to fill in the hole. Right? And that's where this stuff comes from. And part of the stuff that they secrete is more jungle gym. It's like the kids aren't satisfied with the little dome, and they want to make a larger dome, or they want to make another dome nearby, and they can actually spit out more jungle gym, right? And so these cells right here are manufacturing additional matrix, and that's what's deposited here. And the primary matrix that they lay out is collagen. And so you can see this cross-linked triple helix collagen architecture, and so that's what they're manufacturing. So they make more matrix, more jungle gym, and they make more cells. So the analogy, bless you, would be the kids on the jungle gym replicate, and I don't mean weirdly, like they just like all of a sudden one kid becomes 2, and then it's like 2 kids become 4, and then they're spitting out more jungle gym. Like that'd be like a teacher's nightmare, wouldn't it? Like we started with a class of like 35 kids, and now I've got 70 before recess is over. Okay, so that's what's going on in this particular site. Okay, we've got matrix production, and we have cellular replication or divisions. How do they signal? They signal in all sorts of different ways, but now we're looking at this picture where we're looking at a cartoon diagram of a cell. Here's the nucleus. And these integrin molecules are connected via struts, kind of like in your attic, but we call these cytoskeleton, actin cytoskeleton. And so the collagen, the triple helix collagen, is out here, and the cell, the fibroblast, binds to the collagen fiber. So that's this interaction right here, which is this interaction right here, which you can see how far away it is from the blood vessel. And as it interacts, it decides to detach this side, grab onto this side. It might express an integrin molecule over here and grab part of the matrix on this side, and that's how this cell is going to migrate this direction, right? And every time there's an interaction here, the nucleus sees it, because these are physically attached to the nucleus. So intracellular signaling through integrins are directly connected to nuclear activity. And so if the nucleus goes through a proliferation state or a differentiation state, or says, let's detach here and let's attach over here, that's how the cell is actually going to migrate. And that's how that cell crawls through that matrix environment. So we've got cell stuff happening in a wounded tissue. We've got the cells replicating, going through the cell cycle. We have them manufacturing additional matrix proteins, primarily collagen, to fill in this gap. So we have new collagen matrix, we have new cells. Well, the other thing that we need is we need new vascular supply, because as you're doing this, you're needing nutrition, right? You need oxygen, you need ATP, you need nutritional elements that come from the bloodstream, and then you also go through this metabolic process, so you have to remove the waste products. And so in tissue healing, one of the most critical components is it vascularized? So case in point, how many athletes do you have in the room? Even if you're kind of like, more of like the weekend athlete, or you don't have to be like, sponsored or anything like that, okay? Seriously, just like 5 athletes, that's it? Everybody else, what are you guys, gamers, or what do you do? Okay, all right. Nobody goes for a jog, or, okay, this is kind of sad. Okay, all right, more hands are going up. How many of you like jog to the fridge? Yeah, awesome. Okay, that counts. I mean, because you can totally tear your ACL doing that if you're not careful. Okay, so let's do the ACL tear. All right, athletes have an ACL tear. They have cartilage damage, and then you typically hear, oh, well, sorry, that cartilage isn't going to heal very well. Do you know why cartilage doesn't heal very well? It has low blood flow. It's very poorly vascularized. Okay, there's two types of cartilage. There's red cartilage, and then there's white cartilage. Which one do you think is slightly vascularized? Oh, wow, you guys are brilliant. Red, okay? Red cartilage is slightly vascularized, but it's kind of on the edge. And the cartilage in the middle is more white, and that's why a cartilage tear doesn't typically repair very well. So blood flow is absolutely critical to promote wound healing. Okay? So, angiogenesis, the development of blood vessels from pre-existing blood vessels. You can see in this picture how they sprout and then how they mature. And if you remember back to our macrophages and inflammation, they release a compound known as VEGF, vascular endothelial growth factor, right? And this is extremely angiogenic. It stimulates this process to happen very aggressively. And that's really important as we see in this particular picture. So if we could vascularize cartilage, we would put orthopedic surgeons out of business, okay? But because it's a very difficult task, orthopedic surgery is a really good field to go into, okay? Because more and more people are tearing their ACLs on their way to the refrigerator than ever, okay? So here we go. Let's talk about healing. This is healing of a wound. This is a thin wound. This would be a situation known as healing by first intention. A thin wound, okay? You can kind of appreciate the thin sliver of the wound. In a 24-hour period, we can appreciate there's the first responders, our neutrophils. We've got a clot that has formed. We've got a little bit of a scab. There's a little bit of a bump on top. And we see the underlying vasculature that you can't really tell in this picture, but it's slightly vasodilated. 3 to 7 days later, we see that the clot is starting to resorb. We've got mitotic activity that's happening at the level of the stratum basale, because that's the regenerative layer. We've got granulation tissue here because we've recruited macrophages. So these cells now are macrophages. They used to be neutrophils. Those are our second responders. And you can appreciate that the vasculature is more aggressive to bring in a larger blood supply. And then weeks later, we see that the vasculature, instead of angiogenic, now it angioregresses. It goes a little bit back to where it was because you don't need it anymore. And the scab is gone, the clot is gone, and even the amount of macrophages that are present is subsided. And there's a little bit of fibrous tissue here, known as the fibrous union, but not nearly as aggressive as what we see in second intention. So this is a wound, in contrast, that's healing by second intention. So the wound is bigger, right? Larger. You can kind of appreciate how we've drawn here. We still have our neutrophils that come in. We see a more aggressive angiogenesis response, and we see a more robust response with the number of tissue macrophages that come in. And then, of course, in wound contraction, we have much more fibrotic tissue that's present in healing by second intention. And also at the surface, you can see there's a little bit of a non-aesthetically pleasing result because it hasn't plumped up to the same level as the nearby tissue, and that's where you can tell that there's a scar. In some patients, it actually goes the other way, and we form what's called a keloid. We'll talk about that here in a second. Questions on healing by first or second intention? Okay, some of these kinds of pictures, I'm not going to point out exactly which ones, but they might show up on some of these quizzes, okay? Like on Friday. So here's an example of a pressure ulcer on a foot. We're getting into some, I mean, they're not too bad. They're not too bad. We're getting close to, well, there's a nasty one. That's a little down. All right, we still got some time to warn you. I think I have warning hints in here. This one isn't too bad. So this is a pressure ulcer. And you can kind of appreciate where that pressure wound is sitting. If we look histologically at a pressure ulcer, this isn't the same one, but an example, you can kind of appreciate, here is the stratum corneum on either side. You can see this giant gap. This is all granulation tissue that is not resolving. If we look at in panel C here, we've got healing that's taking place. So we're pretending that this wound is actually going to close. So this is what it looks like to start. Here is at some phase in wound closure. And the phase here is where we've got layers of granulation tissue that's formed. I'm going to show you where those are. This is a hematoxylin state section. This one is as well. This is a little higher magnification view, but you can appreciate that the epithelium is completely closed over, right? So now this wound is healing over, which is good. If you look underneath the surface, you see this circle right here. You see sort of this oblong shape, this circle, this oblong shape, that circle. Those are all blood vessels. Those are all blood vessels in the phase of granulation tissue because of the macrophages that were releasing VEGF to make new blood vessels via angiogenesis. So this is what granulation tissue looks like. Doesn't it have kind of a granular looking appearance? That's why it was named that. Okay. If we continue on in the healing response, again, this would be that pressure ulcer from going into full healing. Now you see the situation of complete reepithelialization. You even have a functional stratum corneum up top. You can see a little bit of this is sloughing off. That's what it normally does, the stratum corneum, the superficial most layer of the skin. And if you notice in here, do you see kind of these bands of more pink staining there and then right here? Do you guys see that? So there's a little bit of an area here and a little bit of an area right here. And the pink is eosin, and collagen takes up eosin aggressively. So the larger or the more pronounced the pink staining, there's more amounts of collagen that's been deposited there. So you can see this is the fibrotic scar here as well as here. More pink tissue, indicative of a little bit of fibrosis. I would say this turned out pretty good from a histological standpoint. So again, on healing, if we have wound resolution or wound repair, it's a spectrum. It's never one or the other, right? I mean, think about this. We have all of these terminologies in biology, but the body doesn't care what we label it, right? So it's just sort of a spectrum. So is there going to be ever a situation that's completely one and not the other? No. But it might be like 90%, wound resolution and maybe only 10% scarring or healing. Or maybe it's 95-5. This looks pretty good. I mean, I would be pretty happy with this kind of outcome. So we lay down new collagen. What happens if we lay down maybe a little too much collagen? Okay, this is a keloid. Excessive collagen deposition in certain patients that are predisposed to keloid formation, and you're going to see something that looks like this, where this doesn't stop at this level. It just kind of keeps laying down more collagen than really was necessary, and you get a little bit of a raised bed of collagen. Okay? And certain individuals are more predisposed to keloid formation than others. Histologically, what does it look like? Well, you can kind of appreciate kind of the raised surface instead of flat, so this is that raised surface, but the collagen by the arrows, these patches of more aggressive pink collagen staining. Make sense? So if you compare and contrast this keloid histology to this histology that has maybe a little bit more fibrotic collagen than what you want, but it's not as aggressive as what you're seeing right here. Make sense? Okay. Questions so far? Okay, cool. So we're going to transition a little bit into what happens if we decided that we want to leverage stem cell technologies in healing applications. Okay, because you're seeing a lot of this now in the field. This is being used very actively today. And there's a number of different sources of these stem cells, and we've talked about this in the past, but we're going to characterize it a little bit more right now. So we've got a chart here. And this chart is a pretty good chart that I borrowed from another publication. And so you can see embryonic stem cells over kind of in the far left in the middle, and then we've got adult-derived stem cells that are shown over here on the far right. And that's one of the first questions that I end up getting when we're talking to a group of people and when I'm lecturing on stem cell technologies, is they want to know, well, which technologies are embryonic and which ones are not? And mostly not because really the public understands what embryonic means. They just know that there's something bad about embryonic, meaning probably babies died if it has anything to do with the embryo, right? And so there's ethical concerns. And so that's why it's important to me to make sure that you all understand what the differences are, because not every stem cell is the same. Now, embryonic stem cells, we've got two different categories. We either have in vitro fertilization or we have nuclear transfer. And in both of these situations, with the embryonic nature of it means that you actually do have to destroy the human blastocyst. Okay, so the problem ethically with either in vitro fertilization or with nuclear transfer is you've got the destruction of the human blastocyst early in the phase, just post-fertilization, because you actually have to use that tissue. However, the advantages of embryonic tissue is they are what's called totipotent. Okay, totipotent. So that term is not even listed on the slide, but totipotent. This term means that this tissue, this cell, can differentiate or it can change into any type of cell in the body, which makes sense. If you start with an egg and it's fertilized with a sperm, you've got one cell that now becomes dividing into two, and that one cell that goes into 2, and then 4, then 8, then 16, 32, so on and so forth, becomes an entire organism. So one cell turns into many cells that become skin, hair, nails, muscle, neurons, everything. So that's totipotent. You have the ability to develop an entire organism from one cell. Okay, so what are some of the drawbacks other than the ethical concerns? Well, in the field, we've stopped doing a lot of embryonic work. Not necessarily because of ethical concerns, in my opinion, but mostly because there's too much power in the embryo that humanity doesn't know yet how to control. And so we've sort of moved away from it because most of the time when you use embryonic stem cells, it doesn't turn out the way you want. You've got the ability to become anything, and you get what's called teratomas. And so you get these masses of tissue that have, teeth in them and hair. And we don't know how to control all of the technology or the science that's present with an embryonic stem cell. So you can see on the first column on the far left the risk of creating teratomas. That is an absolute high number on the disadvantages. Like, it happens more often than not. And let's be honest, in the field of science, do you really think that scientists abandoned embryonic stem cells because they worried about the ethical concerns? Or were they worried because it wasn't working? Right? I mean, you can answer that question for yourself. But again, and it's more of a philosophical conversation of why isn't it working? Well, I don't know if humanity is really supposed to be playing over there. And again, that's more of a belief thing. But we're not doing a lot of that anymore as a field, which for some of you might say, oh, that's great for ethical reasons. Others might say that's great because it wasn't working. So whatever you decide, we're kind of there where we're not doing a whole lot with embryonic stem cells. Much of the research today and much of the technology today is over in the category of adult stem cells. Now, adult-derived stem cells have a number of different categories. And we talked about this kind of in the first time we were together. When we're with adult, we can have young and we can have old. Okay? And young sources could actually be what we call neonatal. And so you can get stem cells from placenta, umbilical cord, right? You can get it from cord blood. And you can even get it from neonatal tissue, such as throwaway tissue like foreskins, okay, from circumcisions. What? talk about this a little bit later. It's throwaway tissue after a circumcision. Same with placenta, throwaway tissue, umbilical cord, throwaway tissue, right? So the field has gotten pretty creative about what human tissue that's young, that's discarded, might actually have value, okay? Kind of a cool concept in a recycling sort of way. And then old, sorry, but all of you are in this category with me, okay? Really, neonatal is within the first few weeks of life. And then after the first few weeks of life, you become old. So this would be bone marrow. Okay, this would be circulating blood now, not from cord blood, but just circulating blood out of your body. There's a product on the market that people refer to as what's called PRP, platelet-rich plasma, where you do, this was made popular by Kobe Bryant. Kobe Bryant had a knee injury years ago, and he actually went to Germany to get this procedure done because it wasn't being done in the States. Now, this is a long time ago, actually, years ago. Years is still accurate. But he was in his 20s, okay? He's retired now, so obviously this was a while ago. When you do a blood draw on the patient, you spin down the blood, you get what's called a platelet-rich plasma fraction of the blood. And it's rich in growth factors and cytokines. And then they draw that out, and they injected it into his knee to promote healing, okay? But that is still an old technology, okay? In the context. So bone marrow, blood, and then the other one would be fat. Fat derived. This one's quite interesting today, where we'll do a lipoaspirate, pull fat from a patient, and fat has lots of adipose-derived stem cells in it. And so you can generate a stem cell therapy from that same patient using their own fat. Okay, so they have liposuction and they get stem cell therapy. It's like, happy day, right? Then you hand them like a popsicle. Congratulations. So in the adult sector, the only other one that would be on here would be called what's called induced pluripotent, IPSs or IPCs. Okay, we talked about this in that last lecture. That's an adult, where what you do is you take a variety of adult stem cell and then you modify transcription factors to make it think it's more like embryonic. And so IPS or IPC is kind of like the new embryonic stem cell that a lot of investigators are chasing after. Okay? So that gives you kind of this whole spectrum of stem cells in the relationship to where are they being harvested, how are they being used. So here's just another graphic to describe, kind of the different categories. Embryonic up top, adult down low. This one shows a induced pluripotent stem cell, an IPS or an IPC from normal cells as its own category, but really, I would argue it's a subcategory of adult. Because you either have embryonic or you have non-embryonic. You either have adult or embryonic or you have adult. And so the IPS is really a type of adult stem cell that we're now using today. All right, so I want to finish up today's lecture, the last portion of today's lecture. This is a study that we did back in like 2012. We published it in 2014. And we were going after this fat-derived stem cell therapy that we were interested in. And this is a grad student that was in the lab. And so what we did is we took lipoaspirate from patients, and we go through this process where you have the patient, you generate the lipoaspirate, you aspirate and wash it, and you centrifuge and digest, and you get what's called a stromal vascular fraction. This is a pellet that's in this container that came from fat that contains adipose stem cells. And those adipose stem cells have the ability to differentiate into many different lineages. So let me use another term. I said that embryonic is totipotent, and adult is what we call pluripotent. So pluri means many, and totipotent means total. right? So this can become anything, and this can become many different things. So that's one of the big differentiations. But, you know, back in 2012, we focused on fat because we were trying to stay away from this whole ethical conversation. And it was a good idea because that's the direction that the field ended up going. So this is some work where we've electrospun these scaffolds. These are scaffolds that instead of having a gaping wound and putting cells in it and making them make the jungle gym, we thought, what if you have a wound, you supply it with a jungle gym framework, and then you put in stem cells that help promote, you know, the filling in of this. Can we actually close those wounds in a faster way? So this is how electrospinning works. We've got a target that's negatively charged. You've got a supply that's positively charged. This is a high voltage power supply. This is about 25,000 volts. Okay, and it's low current, so it stings, but it's not dangerous. And so the needle is connected to a tube that's connected to a syringe on a pump that delivers it at a certain rate. And in this syringe is a solution that could be really anything that you want it to be. You can fill it with collagen, okay, and that's what this is a collagen framework. You can fill it with collagen and other extracellular matrix proteins like elastin, which is also found in the skin, collagen elastin. Elastin is what gives skin kind of its recoil characteristics. It's kind of the stretchy protein in the skin, and collagen is kind of like the strength of the skin. So we did exactly that. We put in solution collagen elastin, and then it almost looks like spinning spider webs. So I have a video. Let's see if it plays. This is what it looks like in the lab, the system that we built. Let's see if this goes. No. Hang on. Let's see if it's in my folder. 1 second. There it is. Okay, there's the video. So you can kind of see the sort of the spider webs kind of coming down. Play it again. There you go. So you see those fibers that are spraying downwards? Just through that electrical gradient. That's all it is. But the reason to do this, or the reason that we did it, is you can create these architectures, these jungle gyms, if you will, that look like this. Right? So you can kind of imagine a cell wanting to sit into that matrix and hang on to that fiber. So our theory was, hey, let's populate this wound bed with a scaffold that mimics the surrounding tissue, and then we'll put our stem cells in there, and those stem cells will have the ability to pre-populate the wound bed and encourage new fibroblasts to come in. And so this slide is just showing that we create new materials by altering things like flow rate or concentration. This is just flow rate. And so the right image is a slow flow rate, and the left image is a fast flow rate. So a more dense matrix versus a less dense matrix. And this is a picture of that scaffold preceded with adipose-derived stem cells. So these are human adipose-derived stem cells. They came from donor patients that had liposuction. Yes, we collect fat, and then we took the fat and we harvest their stem cells, and then we put them onto a wound healing patch, and then we're going to go treat wounds. Okay, and so I just need like 5 volunteers. Anybody? Oh, really? Are you guys serious? Oh, that's awesome. Can I just get your names afterwards? I'm just kidding. No. So we did this in preclinical models, but we'll talk about here in a second. So this is the study. We did these in mice, and we did SCID mice. Okay, these are severe combined immunodeficient. That's what SCID stands for. And this is important, this SCID mouse model, because it lacks an immune system. Specifically, it lacks B and T cells. And by not having B and T cells, those human stem cells that we put into the scaffold don't get rejected. Okay? So that's why we use this. We did a 6-day study. We electrospun them first, just like we talked about. We had human donors. These were, of course, former grad students. They're no longer with us. I'm just kidding. They were old people that were fat or something. I don't know. We got it from plastic surgeon, so I don't know why they had it. But full thickness wounds in the mice. We seeded the scaffolds. We implanted them, and then Tegaderm is a normal wound dressing that's used in the clinic. And Vaseline gauze, that was our control group. Here's the model, so you kind of create a score of a biopsy punch, and you trim out the little wound. It's a full thickness wound in these animals. This was Hans, this was his study. These are these scaffolds growing in this dish. Control wounds over here. We've got two different scaffolds in the middle and right. And so the take-home message, dim the light so you can hopefully appreciate these wounds. We traced the inside of the wound that wasn't fully closed. So you kind of appreciate in some examples of control wounds that had Vaseline gauze and Tegaderm, but no scaffold with stem cells. After 6 days, they were, 62 to 76% closed. This was an elastin scaffold, 100% elastin scaffold that was almost always 100% closed, and then a collagen scaffold, and both of these had stem cells in it. So we were pretty happy with these kinds of results. We quantified the data. You can see about 75% in the control just after 6 days, and then we definitely increased wound healing with our scaffolds, with our stem cells. We looked at epithelial thickness, like how thick the epithelium was after the 6 days, as an indicator that more mature wound resolution. So again, another metric that we were happy about is our scaffolds with our stem cells had better epithelial thickness after the first 6 days of healing. So these studies we published in a journal, Advances in Wound Healing or Wound Care, presented that data. Hans went on to medical school, I'll post that paper for you guys if you want to read the whole thing, okay, a PDF of that paper. But where I want to end on today and wrap up is I want to just characterize these, bookend what we were talking about with these 3 categories of wound healing, okay, and then that's where we're going to hit the pause button. So the 3 categories of wound healing that we talked about, now we focused heavily on skin, and we're going to focus on skin again on Monday. As you guys can see, I do a lot with skin, okay? But if you're talking about, we talked a little bit about liver. We didn't talk much about cardiac. I used to do a ton of cardiac work early in my career. And if you're talking about cardiac, kidney, liver, colon, intestine, any kind of tissue that has a wound response, you're going to have these same phases of healing. You've got this early coagulation inflammatory phase, right? Number 1. And we've got neutrophils that come up first, and then we have macrophages that come up next. And then we have this proliferation phase where the fibroblasts start to proliferate. They're recruited by the macrophages that send these signals. And they move in and they start making matrix. And that matrix fills the wound bed. And if it's a lot of matrix over a long period of time, it forms a fibrotic scar. If the matrix itself was intact and you just replenish the cells, you have a much better outcome. We talked about the difference between a damaged tissue that has cells that are traumatized or a damaged tissue that has cells and matrix that are traumatized. And then lastly, we have our remodeling phase or our maturation phase. So if we just look at some of the pictures, again, of what's happening, right? Our coagulation, our inflammatory phase, right? This is from another publication. I like showing you numerous types of publications that say the same thing from different authors. So when you guys get out there and you see stuff, you can synthesize this information. I don't want it to look like only one picture and you never understand how other pictures look, okay? Different publication from Gertner in 2008 saying exactly the same thing. I mean, here's a skin model. You've got this early inflammatory phase where we develop this fibrin clot. We've got platelets that have come in. We've got our neutrophils that they're highlighting as our first responders. Then we have our proliferation phase. Now we've got the monocyte recruiting out into the macrophage, and we start forming, this is, right, scar. This is what this word is, a Latin for scar. We've got extensive vascularization in our granulation tissue that's forming. And then we see the maturation phase where we've got our fibrosis that closes this wound, and we get angioregression. Angioregression is that terminology. After it's angiogenic and you develop new vessels, you angioregress and you bring it back to a baseline situation. Okay? So, this is really James Anderson's graph. This is really, these pictures are really the graph that I opened the whole lecture with, right? Just now these are pictures showing, instead of that, timeline of events. For those of you that are visual learners, maybe the pictures are better than the graph, I don't know. Make sense? Questions? We're going to pause there because the next slide tells me that we're getting into some of the clinical stuff that you guys are excited about. All right, have a great weekend.